Good morning, children. So we are back with cell division once again. Here today what we have done, mitosis in animal cell. I will be now comparing the same thing, mitosis with plant cell. Is it animal cell? Since I have, that is the reason I have drawn here, so that we can compare the diagrams, the phases. Is it all right? So we start with prophase. Prophase, animal cell, as we have already done. Prophase, what do we see? The animal cell, nuclear membrane disappearing, nucleolus, this way we are revising also a bit, nucleolus disappearing, and then the chromatin fibers have started to coil. Is it all right? And we see the centrioles moving apart and the spindle formation is taking place. Is it all right? Here, if we come to plant cell also the same thing. We find with the prophase, we find that the nuclear membrane disappearing, the nucleolus disappearing, the chromatin fibers are now coiling up. They have not formed chromosome. I have not labeled. Please refer to the labeling of the cells, which what we have done in with animal cell actually. Okay, so the chromatin fibers are now coiling up. So you can see the thick chromatin fibers. Where is the difference? There is no centriole. There is no centriole. Is it all right? But we can see the formation of spindles. Spindle formation has started. Is it all right? And these spindles occur by the cytoplasm itself. Is it all right? Cytoplasmic activities lead to the formation of the spindles. But one thing you are not noticing, there are no asters. No asters. Asters should not be drawn. No centriole. No centriole. So I have drawn early prophase. By late prophase, the nuclear membrane has already degenerated, has disappeared. The nucleolus has disappeared. Is it all right? Sorry, I didn't show this. Here, this is the centrioles are moving apart. There are asters and the spindles are elongating. Is it all right? You can see the spindles elongating. All right. Here, the same thing by late prophase in plant cell. This is what I have drawn as a plant cell. You can make out the cell wall. Is it all right? You can see that the nuclear membrane is disappearing. The chromatin fibers now have coiled up to form the shape of chromosomes, and then you see the nucleolus disappearing and the spindles elongating. Now the spindles are elongating but without centriole, no asters again, no centrioles. Is it? So this is early prophase and late prophase compared with, this is the animal cell and this is plant cell. You can make out from the shape also. Now we come to metaphase. Metaphase, you remember, all the chromosomes arrange themselves on the equatorial plane. The chromosomes are duplicated, arranged on the equatorial plane. The centrioles on the opposite poles and the spindles well formed. Is it all right? Here you see the chromosomes are duplicated, arranged on the equatorial plane. Is it all right? There are no centrioles, no asters, but spindles are well formed. Everything same as in metaphase of animal. Same here, but the only difference is here you won't find any centriole, you won't find any aster ray. Okay, now this is what is metaphase. Now let us come to the anaphase part. Now let us come to the anaphase part. Is it all right? In anaphase, I have not drawn late or early, like what we know about anaphase, that the spindles contract, the spindles here, the spindles contract. The spindles contract. This is a duplicated chromosome. The spindles contract, pulling one sister chromatid towards one side and the other towards the other side. Is it all right? So the spindles are contracting and pulling the sister chromatids apart. The sister chromatids are pulled apart. Is it all right? Here also the same thing. The spindles are contracting and the sister chromatids are pulled apart. There is, by late anaphase, you may find a small cell furrow as well. By late anaphase, this cell furrow may appear in late anaphase or early telophase. All right. Now here you won't find any furrow as such. Maybe it be early or late, you will not find. In plant cell, you will not find any. Is it all right? Now, after anaphase comes telophase. Telophase, you find a cell furrow has appeared, all right. There is now they have migrated to the opposite sides. We have done the uncoiling of chromosomes into chromatin fibers. The nucleolus has started forming. The nuclear membrane has started forming. You can see, and there is appearance of cell furrow. Let us come to the plant cell. Now what we have to see. Is it all right? The same thing. They have migrated to the opposite poles. The sister chromatids have migrated to the opposite poles. Uncoiling of chromosomes have happened, forming chromatin fibers. Chromatin fibers, is it clear? They have moved towards the opposite pole. There is no centriole, no aster. Now what will happen? You will find something developing between along the equatorial you will find something developing, a plate-like structure developing between the two newly formed nucleus. In the daughter nucleus you will find, and we call it the cell plate. Is it all right? And the cell plate keeps elongating from the centers. It keeps elongating towards the periphery. Is it all right? It keeps elongating, all right. And by the end, I have not shown cytokinesis in animal cell. We have already done. Now in plant cell, what do we see? That this elongates, finally dividing the cell into two parts. This actually, the cell plate has elongated to divide the plant cell into two parts. Is it clear? This forms the primary cell wall. Again, later on, they will develop their own cell walls later. This is the basic cell wall which has divided these two into two parts. So here, this becomes the cytokinesis. This is what is cytokinesis. Now, let us differentiate between the mitosis in plant cells and animal cell. In mitosis, in this kind of cell division, centrioles are present, no centriole present. Is it all right? Here, what do you see? Do not give the reason that cell wall is present. Do not give. I'll come back to it. Let us finish it. Asters present, no asters are present. Is it all right? Rest all are same. And at the end, you find when the division of cytoplasm starts, here the division starts by the formation of cell furrow, and here the division starts by the formation of cell plate. One thing you see here, it starts from the periphery, the division, see the difference, from the periphery, and it moves towards the center. From the periphery it is moving towards the center, and here it starts from the center and it moves towards the periphery. Cell plate formation. Okay, one more thing. During the exams, in exams you will find one of the stages will be given, either plant cell or animal cell, and you will be asked to give the reason. Sometimes it happens that this kind of diagram is given. Is it? I hope by this it is clear to you. Suppose I talk about a diagram is given like this. Is it all right? And you have to identify. Suppose you have a diagram is given like this. It's neither triangular, not necessary that they will give you only either for plant cell they will give you rectangular shape, or for plant cell, animal cell they may not give you. They may not. Is it all right? Suppose a cell like this is given, okay, and then it is the diagram is given like this, all right, okay, and the chromosomes are shown like this, okay, and then here it's shown like this. Quite possible it can be asked like this: identify the cell, identify the phase. You will say prophase. Why do you say it is prophase? Because the nuclear membrane disappearing, the nucleolus disappearing. Is it all right? They can be still seen, so it is prophase. And then what happens? You are finding the formation of spindles. Spindles are forming. Is it all right? So it is prophase. What kind of cell is this? Is it plant cell or animal cell? This is a plant cell. What is the reason for it? No centriole present, no asters present. In this, no cell wall is shown. So if it is the cell wall is not shown, you just cannot consider that it is an animal cell. Sometimes the diagrams comes like this. Please see the centriole. Is it all right? Or if this particular stage is given, instead of a triangular shape, suppose this shape is given. This shape is given. Is it all right? Not necessary they'll give you a triangular shape. How do you know it is a plant cell or animal cell? A cell plate formation has occurred, and cell plate always starts from the center. Is it okay? So we, instead of saying this as animal cell, this is definitely a plant cell, cell plate formation. Do not go to the shape, do not mention about cell wall. If the reason is asked that why do you think it is a plant cell or animal cell, do not go for cell wall. Do not give reason for like the shape. Is it all right? Shape and cell wall. What are you going to concentrate on? Centriole, astus and cell furrow or cell plate formation. I hope it is clear. Okay. Now, what are the characteristic of mitosis we have seen? Since we have finished with mitosis, what are the characteristic of mitosis we have seen? First thing, it always occurs in the somatic.